So viruses tend to be very weird motherfuckers, and that makes treating them super difficult. They're weird, they have weird metabolism, we don't really know that much about them, so that makes finding and creating antiviral drugs that are effective against viruses very difficult. Now, this isn't to say that there aren't effective antiviral drugs, and in fact, I'm going to be talking about one class of those drugs today that do have a very interesting mechanism of action. So, this class of drugs that I'm going to be talking about is called Nucleoside Analogues, and they're just very interesting in how they work. So, viruses are non-cellular. They do not have a metabolism, and so thus they are lacking all of the machinery necessary in order to replicate. They cannot make more viruses on their own. They need a host cell in order to replicate. So what they do, they enter the cell and basically hijack all of the host cellular machinery in order to stop making host proteins and start making virus proteins. Now, in normal DNA synthesis, the DNA is just made. There's different nucleosides that are added onto each other in a chain. That's the DNA code, the A, the T, the G, the C, all of that. They're just added on normally. But in order for them to be added on, there's two very important groups. So the chain that is grown, the end of that chain must have an OH group or a hydroxyl group, and that hydroxyl OH group binds to the phosphate, so that's just PO3, group on the next incoming nucleotide. That nucleotide is then added onto that chain, and then the next new incoming nucleotide is added onto the OH group on that new nucleotide, etc., rinse and repeat. Now, these nucleoside analogues are essentially the same as these regular bases. They're still the ACGT, but they're missing that OH group, which is very important. Without that OH group, DNA synthesis just stops. No more bases can be added on. Now, since the nucleotide analog still has the phosphate group, it can bind onto the new chain. So that's added on all good. But since it doesn't have that OH group at the end, nothing else can bind to it because there's nowhere for it to bind. Now, since DNA synthesis is stopped, that DNA can no longer be transcribed and translated into mRNA and proteins, and thus the viral, the whole virus, cannot be made because it needs its full genome, which it doesn't have because it was stopped during synthesis, and it needs its proteins because those proteins make up the capsid around the outside of the virus, as well as any important special proteins like fucking reverse transcriptase that aren't normally found in the host cells. So, by taking these nucleoside analog drugs, it just halts the viral replication in its tracks. It doesn't make any new viruses. No more new viruses are made, so the infection can no longer spread, and the existing viruses just wait in the host cell in order to be taken care of by the host immune system. And I think that this is a just very interesting and elegant way to take care of viral infections. Now, obviously, this doesn't work all the time. There's certain cases when it just won't, and you have to do different antiviral treatments. But this is just a good solution, and I think it's just very interesting in how it works.